Good day. First of all, I would like to thank the APASTB committee for giving me a chance to present my presentation with title The Current Perspective of MSc Extended Messages of Regeneration. My name is Cynthia Sartika, representative from Indonesia. So to start my presentation today, here are the contents of my presentation. MSc EV biogenesis, isolation and characterization protocol, preclinical and clinical study, and the challenges about MSc EV. MSc therapy, which has been explained from previous speaker, has gathered a great deal of attention for the last few decades in treating many diseases and injury. Initially, therapeutic efficiency of MAC was associated with their ability to migrate and replace the injured tissue. However, the insight of MAC therapeutic potency is shifting to the paracrine effect of its secreted factors, represented in blue circle. This secreted factor contribute to several beneficial effects such as long-term neuroprotection, increased cell proliferation, improved functional recovery, and so on and so forth. All these mechanisms we know as extended regeneration messages of EV. In March 2021, there are over 5,000 clinical trials using stem cell registered in clinicaltrial.gov, in which more than 1,000 are using MSc. Although there are many published papers about MSc derivatives, the number of its clinical trials are still lacking. As you can see in the chart, there are only limited number of clinical trials using exosome, EVs, and conditioned medium, or we call CM. On this slide, I will explain about EVs uptake that involves donor and target cells. It started off with donor cells that generate the EVs, and then the target cells will uptake the EVs under different mechanisms, such as pinocytosis, phagocytosis, fusion, endocytosis, and etc. The level and mechanism of uptake vary depending on the type of recipient cell, state of activation, and the presence of receptors. The picture C shows, in general, the cargo composition of EVs comprising lipids, proteins, nucleic acids such as RNA, mRNA, miRNA, DNA, etc. This coming slide explains about the role of EV carry cargo and effect of recipient cell. Ragni et al. In the journal Stem Cell Communication explain about the horizontal model transfer of EV as a part of paracrine and autocrine communication network. After EV is released into the extracellular space, then bind to the cell surface receptor, EV which carry the cargoes will initiate intracellular signaling pathway. The cargo are released in the intracellular space and taken up by the endosomal system cell target. This horizontal model transfer show how EVs play roles in communication between cells. The interesting part is that the potency of EVs are affected by many factors. Various culture conditions during EV production, such as cell passages, 2D and 3D culture techniques, hypoxia condition, and additional biological stimuli, as well as donor selection and the recipients. All conditions will lead to different EV characteristics. Currently, researchers have begun to find other functional components in GM for MSC apart from these soluble factors in order to identify the EVs that give therapeutical functions. This diagram shows schematic classification of common methods for EVs, isolation, characterization, and quantification. The panel shows MSC can be isolated from various tissue, and the CM is collected to enrich for EV. In the middle panel, describe the different strategies for EV isolation, such as ultracentrifugation, ultrafiltration, etc. The right panel illustrates the strategies for EP quantification and characterization using electron microscope, nano-tracking analysis, and others. Unfortunately, until now, there is no gold standards for EV's production and characterization. Here I present the characterization result of UC-MSc-CM that was conducted by our teams. Most of inflammatory factors concentration are higher during 24 hours, shows by the blue bar on the graph compared to 40 hours culture time, while the interleukin-6 has the highest concentration compared to other inflammatory factors. To evaluate the effect of conditioned medium in disease model, we conducted in vitro study by co-culturing synovial fluid MSC from grade 4 osteoarthritis OA patient with UC-MSc-CM. This diagram is divided into two. The left graph shows the dynamic of pro-inflammatory and MMP activities which occur in OA patients. The right graph shows that SF-MSC that has been co-cultured with UC-MSc-GM can significantly reduce the pro-inflammatory and MMP activities within 7 days. From this review, the preclinical studies of MSc have been conducted in several diseases such as kidney disease, acute myocardial ischemic, type 2 diabetes, and etc. The result shows that MSCEV have therapeutic potential in treating the disease. In collaboration with VetStem Institute Pertanian Bogor, we conducted in vivo study in mice to see the safety of UC-MSc and UC-MSc-GM. After 14 days observation, there is no differences in the subject's behavior, weight changes, and the excretion between all groups. The injection of MSc and CM was proved to be safe in acute toxicity. The interesting part was the brain activity, which shows the increasing brain activity represented by blue dot in the image below. It means there was a regeneration process in brain after injection of UC-MSc and CM. Clinical trials of MSc EVs already conducted since 2013, and most of the clinical trials are still ongoing. Clinical trials for graft-versus-host disease and kidney diseases have already completed. Studies reported that using MSc EVs are safe and can reduce the pro-inflammatory cytokines, therefore improving the clinical symptoms. The following slides are a schematic of how EV can be used for therapy. The advanced production allows EV to be stored as off-the-shelf product, so it can be administered directly into the patient when needed. Aside from off-the-shelf products, EVs also can be genetically modified by loading them with therapeutic molecules. Before administering to the patient, Quality control is crucial for both production pathway. Despite the therapeutic success of MECEVs in several studies, there are still challenges such as assessment of pharmacokinetics and safety profile still need to be resolved. Determine the clinical dosage and the toxicity effects. Other challenges are related to isolation and production process for large scale. To conclude my presentation, here are the summaries. These tiny vesicles carry messages in the form of biomolecule to inform recipient cell of current pathophysiological state and direct the cell to respond appropriately. Developing MEC for clinical use presents several challenges to investigators and clinicians. The major challenges are to establish the optimum reliability, reproducibility, and robust technique to isolate and purify therapeutic EVs, and to produce EVs on large scale with good manufacturing practices standard for clinical use. The translation of EV into clinical therapies requires the categorization of EVs-based therapeutics in compliance with existing regulatory frameworks. EV are indeed the extended message of MSc. So to close my presentation, the take-home message is the potency from MSc EVs can do a breakthrough in regenerative medicine. I would like to give acknowledgement to my team in supporting the stem cell and EV research and development, which I believe could give much benefit in regenerative medicine. That's all for my presentation. I hope this presentation can be understood and become an idea for developing MSc for therapy. Thank you.